It is the other thing is like, I guess you're not listening software websites anymore. The software thing is a little bit different. The software ones like but it's like multiple grad students you can see the piece of software, and then it's usually on the whole piece of software on the lab page, and then we'll go back and forth. But that's not really complicated.
Last set of faculty talks. So if you guys can wrap up the poster session or get any final food or anything, please do so. And thank you again to visit our vendors. They've all done a really excellent job showcasing their products. So let everyone know again when we resume the talks.
RNA sequencing. Once we actually have it figured out right now, I'm looking for maybe type of language it is. It's like, oh, okay. I'm not sure.
It's just 20 minutes.
I'll have this very annoying clock. It's hard to get tell, like, they're like behind the data. That's I was like, I think it's really bad. And that was yeah, well, we can look at the microRNA size, so we're using the NAS, so it should be pretty easy to memorize. That's pretty standard. Yeah, mostly microRNA is like nothing too good. Okay. Yeah. Oh, okay. We need at least 20 micrograms. That's a lot. Definitely. Better if it's fresh. It's also
Hi, Julia.
Okay, can I get everyone's attention, please? Okay, it's almost 2 o'clock, and as much as I hate disrupting really vibrant discussions over there in the poster session, we want to give enough time to our new faculty at BU here in the GSI to present their new research. So if you guys can start to come back over to the presentation area. We have 3 talks, about 20 minutes each for this last session of the GSI Symposium. Our first speaker is Dr. Paweł Pyrzycki from the Department of Computing and Data Sciences, one of the newest departments here from the Charles River campus. And they'll be telling us about integrating large-scale genomics data to decipher the role of the non-coding genome in disease.
Yes. Top in there, or if you want something where you can walk around.
Always leverage a little settle. But I'll just quickly start by thanking also for putting this all together. It's really nice to be here. This is actually only my second time I've ever been at the medical campus. So I spent most of my time at Charles River campus, I guess that's what it's called. I actually wasn't familiar with that term until recently. I hear you guys saying CRC or something. I guess that's what that means. So I didn't even know that. I've only been here for 2 months, and the building that I'm going to be in doesn't even actually fully exist yet. I have it here in this picture. This is actually a schematic of the building that we're moving into for the new data sciences, faculty of computing and data sciences. And we're pretty excited to be moving into there basically in a couple weeks. So that's pretty awesome. And so, hopefully we can get all like the people interested in genomics over that building at some point to see it, see a view of the river, all of that kind of stuff. But anyway, so today I'm going to be talking about integrating large-scale genomics data to decipher the role of the non-coding genome in disease. And so my lab is or will be entirely computational, so I develop methods and algorithms for understanding and looking at large-scale genomics data. But I think it's always sort of best to approach things from the view of biology and the biological questions that motivate us to get that data and to look at that data. And so, a lot of people are interested in questions of disease and what drives disease. And a lot of people suffer from diseases with complex genetic underpinnings and that we don't really understand very well, and we can't figure out the mechanisms that drive those diseases. And a lot of it has to do with the fact that these diseases are driven by changes in regulation rather than any sort of change in the genes or proteins themselves, right? So as alluded to in my title, we're really interested in how non-coding variants, right, how changes in your DNA outside of the genes, outside of the coding region, lead to regulatory changes, which is changes in how much a gene is expressed, or how much of a protein is created, rather than some sort of actual change in the gene or protein itself. And the reason that's particularly interesting and also particularly complex is because a lot of these effects are cell type specific, and so this adds a lot of complexity because while the DNA of most of your cells, of all your cells, is basically identical, the regulation is what causes things to be different at a cell-by-cell level. Now, luckily, towards addressing these sorts of questions with large-scale, in genomics, we are living in the sort of age of large-scale genomics data. I'm sure you guys are all familiar with the fact that we have tons and tons of data these days. So for example, the 1,000 Genomes Project sequenced more than 1,000 whole human genomes, and then TCGA cataloged tens of thousands of human tumors. And they not only did this project do DNA sequencing, but they also did a lot of other modalities of data collection. So, things like measuring chromatin accessibility or methylation, these are all things that we sometimes call, maybe not totally accurately, but we call these epigenetic changes. So that means changes other than the actual changes in your DNA sequence. And so we've got, finally, in this era of big data, we have huge studies sequencing thousands or tens of thousands of individuals, collecting different modalities, and incredibly recently, people have, incredibly smart biologists have figured out how to do things like take these measurements for individual cells, which is incredibly useful towards this question of understanding how regulation can drive changes at this per cell level. Now, as a computational person, I think of a lot of this data as like a sort of just like a matrix, right? And so historically, a lot of data has sort of looked like this, where we could collect some set of markers or genes information across a lot of individuals. So for example, you could think of something like maybe like a SNP chip, but also just across genes, right? You could potentially have measurements across a lot of individuals for something like 20,000 genes across humans, right? But now, when we're talking about these sort of genomide, epigenetic, or these other modalities of data, we're often talking about taking measurements across the entire human genome, which is much, much larger than just a set of genes, right? So there's no way to take measurements across the entire human genome such that you're getting information for every single base for every individual in a very informative way, right? We have a huge dimensionality this way that's much greater than the number of individuals that we can measure. And so, adding on top of that, these modalities expand this data matrix to be sort of a data cube where we can have individuals by genetic regions by the modality in the data. And when we're talking about taking these single cell measurements, we also think of this as being measurements for cells. Now, if we could fill this whole cube, that would of course be amazing. We can make a lot of inference about what is happening at a per cell level or per individual level. But realistically, what we're really looking at is a cube that looks like this, where it's actually mostly empty, and we're only getting measurements for some cells in some modalities for some genetic regions, right? And so when someone like me with a background in computer science looks at a matrix or a cube that looks like this, where we have some measurements but a lot of missing data, well, this really looks like a problem that needs computational solutions, something that needs algorithms to be developed to understand what is missing here and to understand the limits of what we can try to understand from this type of data. So to sort of introduce my lab, we sort of look at this question as how we can integrate these types of data across disease and normal to extract as much information from this data as possible. And we look to try to develop algorithms that do that. And so that can often be either looking at whole population level differences between diseased populations and healthy populations. It can be looking at individual differentiation between individuals, the same individual healthy and sick. So this could be, for example, cancer, you can have the same individual. Or looking at cell differentiation between and across individuals. And a major focus of my lab is to develop interpretable models to do this kind of integrative work, meaning not just feeding in a lot of data and getting some sort of prediction at the end, but we want to understand something from the underlying process and relationship in the data when we integrate across our modalities or across our individuals and states. And so today I'm basically just going to talk about one such algorithm that we developed, which is a network-based algorithm for integrating single-cell and bulk data to try to resolve regulatory elements in complex tissues. So I'm sure a lot of you guys have sort of seen this analogy before about what the difference is between single-cell and bulk data. So let's say this sort of fruit pie is the reality of your tissue or your individual, of your food or whatever. And bulk sequencing is sort of like taking all of this and putting it in a smoothie and just like ignoring what all the little pieces of it were, but just blending it all and saying, this is what I've observed. By comparison, these single-cell sequencing technologies, right, are sort of like taking the fruits out of this and looking at what every fruit was in it, but you don't really have a holistic picture of what happened anymore. And you can only, collect some of the fruits, and sometimes you want to collect the bigger fruits, lots of stuff like that, right? So bulk sequencing has some advantages. Right, so one advantage is that you have a lot of data when you do bulk sequencing. So you're collecting the entire tissue sample, you're blending all of it. And so what you're looking for is major differences between something like a healthy state and a disease state. Well, you can just look, and on average, you can see that there's some difference, right? It's called the smoothie or whatever. You can see there's some major difference between your two conditions. And the other huge advantage of bulk sequencing is that a lot of that data already exists out there. People have done tons of bulk sequencing across tons of samples. It's already like out there. But of course, single-cell sequencing has a huge advantage of being cell-type specific, right? This is the only way we can get that information about like what's actually happening in each cell, with the huge drawback that we're getting much, much, much less data. So we're getting very few reads per cell. It's a sort of technical observation. And to make things worse, what I'm really interested in is regulatory genomics, like I alluded to earlier, with that really wide data matrix, right? Taking measurements genome-wide. And so, for example, one type of data that I'm really interested in is ATAC-seq, which looks for areas of open chromatin in your DNA. And the reason open chromatin is really interesting is because your DNA opens up, and that's what allows transcription factors to bind, and that's what often leads to regulatory changes. And so if non-coding mutations or other similar events are occurring in open chromatin, we can be more convinced that they have some functional importance, though no guarantee, right? And so the problem is that when you look at this in a single-cell space, that sort of matrix I was describing before, right? You have your cells by genomic regions, and let's say this is one cell. In single-cell ATAC-seq, you might only end up with 5,000 measurements for that cell across your entire DNA, right? Across 3 billion bases, though presumably bid this into some larger size, but you only get like really like 5,000 good measurements because you need, it's really just a property of the DNA, right? You only have 2 copies of your DNA in each cell, right? Do we have a question or no? Okay, so the goal of this algorithm we've described is to try to combine some of these advantages of single-cell sequencing data and bulk data in order to have high confidence annotations of regulatory elements to cell types. We're going to call it a cell walker for reasons that'll be clear in a second. So this cell walker method has two main stages. It has a network building stage, and then it has an information propagation or diffusion stage. So I'm just going to talk about these two stages separately, and then you'll sort of see how it all comes together. Right, so first we have a network building stage. And what we're generally going to do is we're going to build a network of the cells that are being represented in our sample. And so we're going to have two types of edges in this network. The first type of edge and node will be the ones that represent the cells. So the cells here are represented in green, and we're going to build cell-to-cell edges based on just how similar cells are to each other. And the reason we want this in our network is because it'll allow us to share information across cells because of that problem with them only having a little bit of data in every cell when we did our single-cell tag sequencing. And then our other type of node and edge will be what we call labels. Now, labels could really generally refer to anything, but in this context, I'm mostly going to talk about labels being cell types. And so each label will be some description of a cell type. So, for example, you could imagine a label being, a list of genes that are known to be differentially expressed in that cell type. And then we'll draw edges from those labels to the cells that we can strongly associate with that label. And so some cells might have no edges at all to labels, some cells might have many edges to labels, some might only have weak edges to labels. That's sort of signifying the fact that for some cells, we can't really strongly associate them with a cell type. But if you sort of think about what this means, like imagine if this cell can't be associated to any label, but it's closely related to the cell, then that probably means it's related to that label. And so that's the sort of relationship that we're going to exploit in this model. Now, what's really nice about this sort of network model and this labeling paradigm is that we can have arbitrarily many sets of labels. So for example, we can have one set of labels that represents a healthy state and a second set of labels that corresponds to a disease state. And we can expand this arbitrarily because it's just a general framework for studying our cells of interest in labels. Okay, so that's how we build our network. The second stage is propagating information throughout that network so that we can actually sort of learn something new. And so the way we're going to propagate information through this network is through a random walk. Some people here maybe aren't familiar with random walks, so I'll quickly talk about what a random walk on a graph is. So you can sort of imagine a person starts somewhere on your graph, and they walk along your graph or network proportionally to the weights on that of those edges. And so you could start sort of at any node and end up at any other node. But what's interesting about this framework that we're showing is that you can start at a cell and then at a label, but you can also start at a cell and then at another cell. You can start at a label and end at a cell, and so on, right? So we do a huge number of these walks across the network, and we're sort of observing where walks start and where they end to understand how information can flow through this network from labels to cells, between cells, and also from labels to other labels. Luckily, there's actually a mathematical formulation for this sort of random walk that you could solve, which is very helpful for us, but the main thing that matters here is that after you've done this walk, it eventually goes to convergence because we're using a model called a random walk with random restarts, which means that over time the amount of information decays, and so you're guaranteed to converge to some final state of where your walks end up. Okay, so after you've done all these walks, you end up with something called an influence matrix, or what we call an influence matrix. And we really like this influence matrix because it's a really detailed description of what happened between cells and between labels in these walks. And so if you take any one column of our influence matrix, that tells you how the fraction of walks that started, for example, at the cell that ended at every label and every other cell in each vector like this. Now we can derive a lot of information from this influence matrix. For example, we can take a row here of the influence matrix, and this will tell you how many walks from each of these labels ended up at this particular cell. And that's really useful for something like labeling your cells to a label, to a cell type, right? So you get a continuous vector of scores that describe how similar to each label that particular cell is. We can also use the number of walks that went from each label to every other label to build some sort of cell type cluster to see how similar labels were to each other, because that's how many walks went from each label to each label. And what I want to emphasize here is this isn't sort of just like some sort of hierarchical clustering just on like label information. This is telling us how often walks go from labels through your cells, right, through that network of cells, back to other labels. So it's using that single-cell accessibility to group our labels. You can also use the cell-cell information to do a 2D embedding, which I guess you guys probably are familiar with, but people love 2D embeddings to sort of like give you a sort of picture of how cells are related to each other. And so this gives you a 2D embedding that sort of influenced by what the cell labels were. And actually what's most interesting for us, you can use this vector of cell to label influence to take bulk regulatory elements and map them onto labels via which cells those elements overlap with. So this is a really nice framework, a very interpretable framework for information and how it's shared between labels and cells. Now, we did a bunch of simulations to evaluate how good a framework this is. I probably don't have enough time to really go through the simulations, but suffice it to say that we see that the one parameter we can change is the ratio of the edges, the weights of the edges between the labels and the cells themselves. And actually, I guess what's most important is that if you set those weights to 0, that would be the same as just doing a de novo clustering on the cell network, right? Which is what that's like Louvain clustering is what a lot of clusterings do. They just cluster on just the cell-cell similarity. And that's what this point here would be. And then we can see that as we increase that weight, we actually learn a lot more about how cells relate to each other until you overfit, which is basically you're just saying whatever your strongest edge was to the label, we'll just assign that as your label. We can also see in simulations that this method is really, really robust to having fewer labeled cells, so fewer edges to those labeling nodes. And of course, we see that it's really robust, which is really great, to spiking in noise or reads being dropped out. So that means that you can have really sparse data and it's also work. As a validation, we checked if we knew a ground truth of what cell labels, true labels were using a multi-ohm assay. So like in this case, it's snare-seq, which measures RNA and attack in the same cell. And we assumed that the clustering of the RNA gave us true labels on the cells. Then we also used CellWalker to label based on just the accessibility portion. And we saw we did a really good job deciding the same labels, much better than Seurat. And we were especially pleased with the fact that we were really good at labeling rare cells, which Seurat was really good at bad at labeling. And a lot of this comes from the fact that Seurat, if you guys have used it, tries to build clusters first. It's really hard to build clusters on really, really rare cells. Okay, so what about labeling bulk regulatory information? And so this is really hard to evaluate, but we had a collaborator that had a very particular data set that allowed us to do some evaluation of this. So we had single-cell attack sequencing in the developing brain, and we had labels from this huge project on the developing brain. We had a collaborator that did microdissections in the developing brain, where they tried to get as specific as they could with the data that they were collecting, so that it was like, it couldn't be cell type specific, because obviously you can't just like microdissect cell types, but they could be very enriched for certain cell types based on the region that they dissected. And what we saw was a really strong enrichment in the labels that we assigned to the regions that were in the microdissection, meaning that just using these two sets of data, we were assigning cell types that pretty well corresponded to where the dissection had come from. And then initially I've been talking about our interest in disease, so of course what we did next was then we looked at bulk data from collected in disease states, in this case, or disorders I should say, including ASD and developmental delay, and we could then map those disorders to very specific cell types using this model. So we managed to see that there was a significant enrichment for interneurons in ASD and radioglia in developmental delay in this case. Now, we also applied this model to a lot of other data sets that I won't get into, but it's a really general model that scales well to other domains where single-cell attack sequencing data has been collected. So I don't have too much time, so maybe I'll just quickly say that currently there's a lot of discussion about what is a cell type, and you notice that a lot of our labeling refers to specific cell types or cell type labels, but we're trying to move past that towards not having to talk about specific cell types. So here I can show you can see that if two cell types are really similar, the scores they get in our model are also really similar. Well, we actually know that these cell types are related, and we can apply a hierarchical model to our labels to have that as prior knowledge on our model. And then, when we label our cells, we actually label them through this whole, I should say, we just add this right onto our network. So it's a really simple, intuitive addition to this cell walker model. It's just adding more nodes to the same graph with internal nodes for those new intermediate cell types. And then you can score every cell for every leaf cell type and also every intermediate cell type in your hierarchy. And you can see that this actually allows us to assign labels to way more cells. A lot of these cells, these gray ones were ones that weren't assigned before with just the leaf cell types, but now we're assigning labels based on these intermediate cell types because they sort of look like something in between those two cell types. Last thing I want to say is just a quick note that CellWalker has a full R package that anyone should be able to use and run with an interface that's very user-friendly. So I encourage you guys to check that out. It's on GitHub. It's called CellWalk R, because it's an R. And then I'll just like to thank the Pollard lab where I did most of this work during my postdoc, some of these other labs that I got data and collaborated with. And finally, I want to say, my lab's going to be up here on the 16th floor of this new data science building. I would really love you guys to come visit, check out the view, or come chat science, whatever you guys prefer. And yeah, and come see me. So, thank you.
Thank you very much. Questions?
Oh, great talk, interesting. So in your algorithm, the cell walk, did you arbitrarily set a permutation level, or is that determine how many times it will walk?
For the walking itself,
yes.
It actually doesn't matter because the walks actually converge. So you can do it so there's basically a closed form solution to this problem where you can invert the adjacency matrix and actually figure out what all the walks will converge to in the long run. But then it's very slow to invert the matrix. So we actually did another calculation where we just saw how many steps you needed to take to get really close to convergence. And we found you actually only need to take 5 steps in the graph to get sort of convergence state. Is that what you're asking about? Or is that, yeah.
So it's consistently around that number?
Yeah, it's consistent. Well, the reason it's around that number, and this will depend on sort of the setup, but the way we built our cell network, it's a pretty dense graph. And when you have a dense graph, it takes much fewer steps. There's a term called the diameter of the graph, right? And that determines how long it would take for a walk to go from end to end of the graph. And so if it's densely connected, it's pretty easy for a walk to reach anywhere in the graph. And so then therefore it requires fewer steps to reach convergence.
So when you use the same label and different data sets.
Oh, in that sense.
Yeah.
Yeah. So the label set doesn't affect the amount of time it takes to converge usually. There's some interesting questions there. For example, if there's a really, really rare cell type, which means that those cells are less connected to the rest of the network, that would require a little bit more time to converge, most likely, because the walks can't really get in there. But it should generally be still pretty good to converge, yeah.
Great. Any other questions? Yeah, so here.
This looks really nice, thank you. Is this extensible to multiple different modalities of data? Can you incorporate data from flow cytometry or?
Yeah, so hypothetically, it should be extendable to basically any sort of data that you can do. I mostly talk about it in single cells, or anything where you have cell similarity, anything where you can assign some sort of labeling where you can build an edge from that label to a cell, and then any sort of bulk data that can be mapped to cells as well, right? So this is a nice, easy context because regulatory regions are, you just map the accessibility of regulatory regions to the accessibility of the cell, and then the labels come from single cell RNA. But there's no reason why it shouldn't extend to anything that has those 3 sort of intersecting components. And definitely something I've been thinking about is what kind of other data can we apply this to? It's easily, it's like the easy extensions. That's basically what I do. So if you have suggestions, I'm happy to chat. For data that looks good.
All right. Well, thank you very much, Paolo, for that excellent talk. And move on now to our next speaker, Dr. Angie Serrano. She'll be telling us about KMT2D's role in cardiovascular and neurodevelopment.
Okay, hello everyone. Wow, that was amazing. I'm the kind of scientist who's very lucky and grateful to be able to collaborate with Paolo. So I started my lab this January in the Center for Regenerative Medicine, and the main focus of our lab is to study rare diseases of epigenetic origin. And in this particular case, we are going to discuss Kabuki syndrome, which is an autosomal dominant pleiotropic disorder that basically affects the development of multiple systems and organs. But 70% of Kabuki syndrome patients will have some kind of congenital heart defect, which will mostly affect the prognosis of the patient. And also 90% or even more of these patients will have neurodevelopmental disorders that will impact the quality of life of the patient and also of their families. KMT2D is caused by pathogenic variants in the histone methyltransferase KMT2D, which is known to promote an open state of the chromatin, allowing the transcription of specific signaling pathways and genes. And despite several efforts to try to understand the specific impact of KMT2D during development, specifically in early embryogenesis and in the context of cardiovascular and neurodevelopment, to this day, very little is known about the specific targets of KMT2D during development. So we're very interested in trying to understand this in the context of embryogenesis, early embryogenesis. So the main broad question of our lab nowadays is to try to understand what are the cellular and molecular mechanisms that are underlying cardiovascular and brain phenotypes in Kabuki syndrome. And we're mostly interested in those that are shared in the patterning of these organs, with a goal in the future to pave the way to seek common druggable targets that will ameliorate the symptoms of these patients. So in order to do this, in our lab, we use a combination of two highly complementary systems. One in vitro is the human iPSC-derived brain organoids, and the other one is in vivo system in zebrafish model. So I'm going to discuss today a little bit of how we try to understand the role of KMT2D during cardiovascular development, during neurodevelopment, and I'm going to touch upon some of the common molecular mechanisms that we've been identifying. For the sake of time, I'm going to go pretty quickly over the cardiovascular results, but those are published, so I'm hoping that you will get excited enough to go and check that publication. For those of you who are not familiarized with zebrafish, this is a super cool model to try to understand cardiovascular disorders, because these embryos have the ability to subsist at least 5 days post-fertilization with a very disrupted vascular system, and that is because of the passive diffusion of oxygen. So here in this case, I'm just showing you the protein of KMT2D in zebrafish. In the conserved domains, we have approximately 90% of identity with the conserved domains in humans. So it's a really good model for KMT2D studies. And I'm showing here in this cartoon a lateral view of a zebrafish, and we're gonna focus mostly in this cephalic area, paying a particular attention here in the heart. So here is an embryo at 3 days post-fertilization. In this homozygous mutant for KMT2D, we did not observe any phenotype up to this day. 3 days after is when we just start observing this phenotype. That you can mainly see this vascular abnormality here. The size is a bit shorter, and we can also start seeing that they have also a smaller brain. We wanted to get more resolution about what were the cellular events leading to this vascular phenotype. So we cross this zebrafish line with the transgenic fish line that will allow us to visualize the patterning of the blood vessels. In this case, it's the KDR line, and we can observe with GFP all our blood vessels over here. These are the loops that are going to irrigate the gills. We have over here the eyes, and this is the heart. Underneath all these loops and blood vessel loops, we have the aortic arches, which are of great importance for us because some of these arches are homologous to the great vessels in humans. So we wanted to see if we could validate or mimic what we observed in the clinical manifestations of the patients. When we observe a KMT2D mutant, it's very evident that the cardiovascular system is completely mispatterned. So you can observe how these aortic arches are missing or truncated. And this is showing us a vasculogenesis effect, and also we can observe angiogenesis effect in most of the blood vessels. We perform a series of different experiments that allow us to learn that these endothelial cells are having a behavioral phenotype, that we do not have less of these cells. They are just clumping together, obliterating the lumen of the heart and of the blood vessels. So we perform RNA-seq analysis. In this case, was bulk, so we have that smoothie that Paul was talking about. We did have a very strong target, which is also very known to be a main effector in the decision step between angiogenesis of the thick and the stalk cells to form new blood vessels. And that candidate was not signaling. So we actually wanted to try to understand what were the mechanisms leading to this phenotype. And we went ahead and tested our hypothesis that not signaling that we observed that was upregulated in the mutant, in KMT2D mutants, that was causing this phenotype. So we went ahead and tried a very well-known Notch inhibitor. This is a very simplified cartoon of the Notch pathway, and we were inhibiting right over here, preventing this Notch intracellular domain to go into the nucleus and activate the gene expression targets of Notch signaling. So here I'm showing you something that I already showed. This is a zebrafish embryo at 5 days post-fertilization, and you can observe all these blood vessels. what happened that are mispatterned, what happened if we inhibit not signaling. And to our surprise, and with a very robust quantification, it was very striking for us to see how we partially rescued the volume of the heart. So these endocardial cells in the heart were forming a bit more the lumen that was lost at the beginning, and we started recovering all these aortic arches that were missing or mispatterned. So this allowed us to try to understand KMT2D role during vascular patterning through regulation of NOT signaling. And we knew that by inhibition of NOT signaling, we were able to rescue this cardiovascular phenotype. What was striking for us, and always thinking about what are the common mechanisms driving the development of patterning of this system, is that NOT signaling is also very well known during angiogenesis. So in this case, this is a very simplified example, cartoon of cortical angiogenesis, and NOT signaling here is promoting the self-renewal of these neuronal progenitor cells. So we have NOT signaling, we have self-renewal progenitor cells, and NOT signaling is at the same time preventing this differentiation of these cells. So if we had more NOT signaling in our KMT2D mutant, our hypothesis was that probably this was driving a neurodevelopmental phenotype, promoting more neuronal progenitors. So we went ahead and tested this hypothesis, and here I'm just showing dissected brains of zebrafish at 3 days post-fertilization, because it's when we started seeing the abnormal phenotype. And I'm labeling here in green neuronal progenitors, in magenta are the mature neurons. What's striking at 3 days are two things that I wanted to take home from this picture. The first one is that the size is a bit reduced, which is super interesting. We don't have loss of a structure, but we have just reduced size. And we also observed this persistent population of neuronal progenitor that was here in the KMT2D mutant that then we quantified. So this was aligning with the hypothesis that probably the NOT signaling hyperactivated was leading to this neuronal progenitor phenotype. So here I'm just showing you, again, this as a reminder of how NOT signaling would promote this self-renewal neural progenitor. And since we had this nice essay of inhibition of NOT signaling, we decided to assess if we inhibit NOT signaling in a wild type, then we would be pushing all those neuronal progenitors to differentiate into neurons. If we already had higher levels of NOT signaling in our mutant, then this differentiation would not be that efficient. So in our experimental approach, we tested different concentrations of this NOT inhibitor, and we collected at 2 days because we wanted to catch a molecular event before we observed the abnormal phenotype in gross morphology. So no surprise, at 2 days, the relationship between neuronal progenitors and mature neurons is not different. At the top, we have the wild type, and at the bottom, KMT2D in the control. But when we add 50 micromolars of DAPT, it's astonishing how we are able to push all these neuronal progenitors into mature neurons. But in our mutant, we still have a persistent population of these neuronal progenitors. Following with our rationale, then if we increase the dosage of this inhibitor, we would finally push all these neuronal progenitors to differentiate. And it was very astonishing for us because it was a proof of concept that the phenotype, neurodevelopmental phenotype in zebrafish due to KMT2D mutation was this differential ratio between mature neurons and differentiated neurons, and that NOT signaling was actually responsible of this persistent population of neuronal progenitors. We were super excited about this, and we wanted to see how can we translate this and to understand what's going on in a human brain, to see if we can identify what are the specific neuronal populations that are affected in Kabuki syndrome patients. To do that, we moved into a model that will allow us to ask those questions. So we started this generating human iPSC derived brain organoids in a protocol that has been published recently from one of our collaborators. And the goal is to generate these organoids from patients and also from a CRISPR engineered isogenic line. And those are the results I'm going to share now. And here I'm just showing an example of how these brain organoids look at 1 day, 7, and at 15 days. And in this case, it would be similar to 1 month-old organoid. This is just a confocal picture of these brain organoids of one of these sections. And I'm using exactly the same staining method that I use in zebrafish. In green, you have neuronal progenitors, and in magenta, mature neurons. And again, in these mutant organoids, we have reduced size. And we can already observe with this SOX2 labeling that we have more of these neuronal progenitors population that, of course, we quantified through 2 different methods to be confident of these results. So we had this increased and persistent population of neuronal progenitors. We know that they divide. We wanted to understand particularly at what point these were aberrant. Are they able to divide and then they're dying, or are they actually failing to exit the cell cycle? So we performed an EDU pulse experiment. So we added the EDU that was included into the nucleus of dividing cells. And in a normal scenario, these cells would divide and the EDU would dilute, while we have both of symmetric and asymmetric differentiation with mature neurons with diluted EDU. Our hypothesis was that in the mutants, this was not happening. These cells were not exiting cell cycles, so we would have higher concentration of this EDU in KMT2D mutants. As you can observe in this picture, in yellow I'm showing the EDU staining. In the KMT2D mutant brain organelles, we have higher concentration of EDU in each of these cells, and we have more of these dividing cells. So we were actually corroborating that these neuronal progenitors were aberrant. They were not able to exit cell cycle. Now the question is, what neuronal population are we missing because of that? What's lacking in more mature organoids? So this is something that Carly Golden did during her rotation in our lab, and started assessing what are the cellular the markers that can give us some clue about the neuronal population that are missing. And here at 1 month organoid, you can already see that this very iconic TBR1 positive marker of post-mitotic neurons is completely missing in the KMT2D mutant. But then it's logical to think probably this marker will come up later at stage. It's just developmental delay, and we will observe it. It happens that at 5 months, we not only have 0 expression of this marker, but also these cell populations. In this case, I'm showing here intermediate progenitor and some of the excitatory neurons, they are missing. So now we have the ability to see particularly the neural population that's been affected by this mutation. We took an approach to validate this through FACS sorting. And in this case, we used this supervised method for clustering, in which through these markers, SOX2, TAPI, forward and side scatter, we were able to give a different characteristic to each of these cell populations. And you can observe here, based on this marker, in the mutant, we are lacking some of these. This is basically a way of saying that we can put each of these cell populations in some kind of stage of cell cycle division and neuronal differentiation. And as you can see here, in this other way of visualizing this, in y-axis we have the cell count, and in x-axis we have the fluorescence of TBR1, which is the marker that we're interested. You can see how in our mutant we are missing these key cell populations that have higher expression of TBR1. So is this also something that we can rescue with inhibition of non-signaling? We proved that in zebrafish, but now we have the ability to specifically identify and pinpoint what's the neuronal population that is being regulated through the KMT2D and non-signaling axis. And we took the same approach. We inhibited that with DAPT, and it's very astonishing to observe here in this KMT2D mutant that was treated with DAPT, how we were able to recover the cell population that had mid levels of TBR1 expression, but no, not that one that had higher expression level of TBR1. So this is exciting. Yes, we can recover this one cell neuronal population that is immediately affected by this, but there are many open questions that we still need to solve. So it makes sense. KMT2D and non-signaling is not the answer for everything during cortical neurogenesis. So this is opening the doors to try to understand what else is missing in this puzzle. So we have thousands of open questions. I'm just going to state two of them. And in one of them is KMT2D regulating in a time and cell-specific manner cell populations that are going to divide? And what's the hierarchical order between KMT2D and the signaling pathway that is regulating, and even TBR1, which is a transcription factor also known to regulate different pathways? So instead of keep digging into the single-cell RNA-seq data, we took a very simple approach. We asked in an iPSC mutant KMT2D line, what are the pathways that are affected? So in this plot that is not complete for some reason I cannot explain, but we identified it was just a set of pathways that were affected in a single nuclear attack seat in our KMT2D mutant that were higher for KMT2D or higher for the wild type. And luckily here you can see WINS, which is one of the pathways that caught our attention. And you can observe here in this heat map how in KMT2D, Win3A signaling was already had higher accessibility. So when we saw not a Win signaling and we knew that we have a not signaling effect, I kind of started hyperventilating because the Win signaling regulation during neurogenesis is very controversial. There are so many unknowns in terms of lineage specification that I said, well, we need to get to the specific of this. So now Carly's project is going to actually tackle these questions, and she's already started investigating the effects of Win signaling and how is a Win being affected in both KMT2D mutant and in wild-type organoids. And you can observe already here that in oligodendrocyte precursor cells, which are known to be affected by Win signaling, and this Win signaling actually the one that will help to the decision step of glial lineage or neural lineage, we can already observe that in planar cell polarity Win signaling molecules, we have a higher normalized enrichment score here in KMT2D mutants. And when we talk about planar cell polarity, we think about migration, cell migration. And we also know that Win signaling has been involved in the glial guided migration of neurons. So let's look at some movies and let's see how if this is affected in our KMT2D mutants. So here I'm just showing a wild-type brain organelle, and you can see these beautiful neurons, how they're migrating in this radial pattern, while in our KMT2D mutant, we are failing to do so. So this is exciting, and it's opening many doors and leaving more unanswered questions than answers, but that's what we're here for, just to answer these open questions. So as a summary, and with 50 seconds left, I just want to highlight that we were able to show in both of our models that KMT2D mutations develop as, and show as a higher proliferation of neuronal progenitor cells, and that we also know that not signaling is upregulated and probably preventing this mature differentiation of neurons that are TVA positive cells. And we also show that if we inhibit not signaling, we're able to rescue some of these mature neurons. And now we have this open question of whether Win signaling is also impacted specifically in the oligodendrocyte precursor cells, for which we will need to go to later stages. And with that, I would love to thank a lot the Mason Center for Regenerative Medicine, which is a wonderful place to work, and also all the members of the lab that were brave enough to join us in a super new lab, and of course the collaborations in mutants. And as Paul, we're also hiring. Thank you.
Fantastic talk, Angie. Any questions? I'll start off with one. What's the chromatin state in these mutants? Since KMT2D is a histone methyltransferase.
Yeah, so I'm gonna be completely honest with this. When I started this project early in the 80s, I was convinced that all these phenotypes were independent of chromatin methylation, but we did perform single nucleotide in many samples. From the ones that we analyzed, it's quite striking that at 5 months, the chromatin state is almost untouched. Between mutant and wild type, there's probably one cluster, so much that I thought I really messed up the experiment, but then we had triplicates, and then we did in several developmental stages. So there's a very small change in the chromatin accessibility that translates in a huge transcriptomic change, which is exciting from the therapeutic point of view, but
very bizarre.
But head scratching.
Yeah.
But actually, there is another disorder, CAT6A, and we shared this with another researcher. She found exactly the same in that disorder. Chromatin state is small changes in chromatin, big changes in transcriptome.
Wow. Any other questions? Could be that people are getting a little exhausted, so we're going to move on to the final talk, definitely not the least, but the final talk by Julia TCW from Department of Pharmacology and Experimental Therapeutics. And she will be talking about cholesterol and matresome pathways dysregulated in Alzheimer's disease brain astrocytes and microglia.
Good afternoon, everyone. My name is Julia TCW from Pharmacology. Thank you for the introduction and invitation. I was here just over a year. So this is the big picture overview of what we are doing in general in our laboratory. We are using stem cell iPS-based model for studying Alzheimer's disease, especially for functional genomics work. And the left side is more about the computational part that we are working on. I train on computational genomics for Alzheimer's disease, and we work on identifying through the 5 mapping process to identify the target genes that regulatory element and target SNP, and also from the network analysis and downstream functional analysis, we can predict what could be happening in a genetic side, and further validate with using human iPS cell-based model, both isogenic and population model, and converting genome edit by CRISPR, and also differentiated multiple brain cell type in both 2D and 3D dimensional. And also since glial cell in vitro, they are pretty finicky cell types, so we injected them into the brain to understand the aging as well as the amyloid challenge. So today I'm going to talk about our most recent publication in a cell about cholesterol matresome pathways that identified in these glial cells. So the well-known feature of Alzheimer's disease are amyloid plaques and tangle, as evidenced by O's Alzheimer's. And he also reported back then in the lipidosis phenotype with the non-red staining, which represents intracellular lipid accumulation in cell around the plaques. In Alzheimer's AD brain have shown that the amyloid around plaques are GFAP positive astrocyte. And more recently in mouse model, they have been shown that the cells around amyloid are microglia, and they express a specific set of gene called DAC, disease-associated microglia. And one of the most significantly upregulated gene is called apolipoprotein E, called ApoE. That's lipid gene, and that maybe it's associated with these lipidosis phenotype. So this is our AD GWAS. GWAS study have confirmed that ApoE is the most significant risk factor for late onset Alzheimer's disease. The significance is way above the actual y-axis. ApoE has a 3 isoform, E4, E3, and E2, and homozygosity of E4 increases risk over 15-fold in Caucasian. ApoE is a missense mutation determined by the TUSNIT at amino acid 112 and 158 that converting cystin to arginine have determined the 3 different isoforms, and it has a structural difference between the isoforms. Once it's translated, it wraps around a lipid particle and exports to the extracellular space is known as a canonical pathway. And within the AD patients, nearly 40%, even recently reported 50% carries ApoE4 allele. And where are they expressing? It mainly preliminary expressed in astrocytes, and upon activation and Alzheimer's challenge, they are expressed in microglia as well as neuron. And even a comparison with the mouse and human, these are the AD GWAS nominated genes. And ApoE is mainly produced and secreted by astrocytes, and even in mouse model, it shows that it's upregulated in APPS1 mouse model. So in this paper, we have tried to understand that what is an ApoE4 risk in brain cell type using these 3 different models: human iPS cell model, postmortem AD brain model, and mouse model. And we use a human iPS population as well as isogenic model, and differentiating 4 different brain cell types. In postmortem AD brain model, we utilize a large brain bank, including Mount Sinai brain bank as well as Rosemont data. We sort out the ApoE4 brain patient and 3-3 and measure the different brain cell type and further deconvolute to compare 4-4 versus 3-3, just like in the cell model. And to compare the species difference between mouse and human, we utilize a target blitz mouse, which carries human ApoE43 genes instead of mouse genes, as well as ApoE knockout and control, and purify the primary microglia and astrocyte, and perform global transcript analysis. So I'm going to talk about the genetic aspect on these collected iPS cell lines. So we screened for the for 2 ADRC cohort and mapped for the downstream genome human reference with the multiple ethnic backgrounds. And these red colors are ethnic group, and this part is a Finnish and the rest European. And we selected 6 and 7 ApoE44 versus 3 individual, and Caucasian and mixed gender, and different diagnosis of stage of AD and age match control. And to confirm that there's no other genetic risk variants affecting to the model other than ApoE4, we have generated AD polygenic risk score using GSI Symposium. As you expected, if we with the ApoE4 genotype, these are the most risk who carries the ApoE4 allele. And when you remove this ApoE4 SNP, and it's randomly distributed, so we selected 39 out of roughly 45 lines, and selected these red, pink, yellow color one for the isogenic correction. And keep in mind that these are not monogenic. The Mendelian, this is not Mendelian type of mutation, but a genetic risk factor. So, meaning that there are people who actually don't carry the ApoE4, don't develop the Alzheimer's disease. And this is one of our colleague, Lindsay Farr's report that there are ApoE modulators that modulate the ApoE gene in terms of expression. So, and also there are another group in Jeffrey Venn's group showing that the different ethnic backgrounds, the African American carries a different ApoE haplotype that also affect the ApoE4 risk, basically reducing the ApoE4 risk. And also that when you look around the ApoE locus, there are so many genes in addition to the ApoE risk. So what we did is that we further look at the using SNP data, selecting ApoE locus, we look at the haplotype around this ApoE locus, and we identified it. These are, remember, these are all Caucasian background, and when we reference the downstream genome from common gene to the rare variants, the lower number is a common and higher number is a rare variant, and our lines are spread out across the common to the rare. So suggesting it was super diverse background. We didn't see mapping background. And so strikingly, when we exclude ApoE4 SNP and cluster the rest of it, and I found a very interesting result that ApoE4 haplotypes are segregated based on a genotype dependent manner. So not only ApoE4 SNP, but also haplotype influences the predicted to downstream pathway differences. And also think about when you generate a CRISPR editing line, and when you create when you added a new 44 to 33 or 33 to 44, you're actually creating a novel haplotype that carries an ApoE4 haplotype in an ApoE3 background or vice versa. But despite that, to understand that how ApoE4 SNP influences to the downstream, we generate CRISPR edited lines converting 44 to 33 as well as 33 to 44. So we have total generated 30 isogenic line in addition to the 2 individual from a knockout. And the difference in these isogenic line is that we have corrected, uncorrected, and knockout actually from the same CRISPR process, not using the parental line versus corrected one. Then with having roughly 43 lines, we have differentiated the 4 different brain cell types, including mixed cortical culture, which is composed of excitatory, inhibitory, and inhibitory GABAergic neuron, anticholinergic neuron, as well as a little bit of astrocytes, and pure astrocyte, microglia, and endothelial cells. And these are the cell type marker confirmation. And then perform our sequencing analysis. A major finding that we have was from the glial cell, both microglia and astrocyte. ApoE44 has the this red color one are lipid-related pathway, including cholesterol biosynthesis and lipid biosynthesis, and also the ApoE44 microglia has negatively regulated lysosome and H-mediated lipid transport. And in a mixed cortical culture, since it has a variability between the differentiation, so we measured all the neuronized and astrocyte proportion and covariate for that, and then identify that this pathway called the matrosome, a matrosome-associated gene, and are enriched, and these other cytokines and ECMs are part of the matrosome. And matrosome is the biggest one, and within that there are core and core matrosome as well as associated core matrosome-associated pathway. So next we asked that what is the up or down regulated gene within these red color lipid as well as matrosome. So we found that the genes that enrich in cholesterol biosynthesis predict in microglia predicts to upregulated cholesterol synthesis, and lysosomal pathway gene, including LEM1, LEM2, MPC1, and 2 genes are downregulated. It predicts to increase of accumulation of cholesterol and decrease of catabolism of lipids. And genes that enrich in an HCMT lipid transport, including ApoE and ABCA1 and ABCG1, are downregulated. It predicts to a decrease of catabolism of lipids, and it suggests that it has a cholesterol dysregulation in an ApoE44 microglia, and also in astrocyte is again showing the synthesis of cholesterol. What about the matrosome pathway? We identified the 3 major functional modules associated with the matrosome pathway in ApoE44 mixed protocol culture, and one of which is the chemotaxis, upregulation of chemotaxis gene, and increase of these genes involving an increase of activation and inflammation, increase of third set of genes involved in a synthesis of lipids. Surprisingly, when we perform these postmortem AD brain data, regardless of any trait of the diagnosis, when we compare AD versus control, the most significant upregulated genes or enriched genes are core matrosome and matrosome pathways, and other ECM and cytokine. And we then ask about where is this genotype and cell type is this signal derived from? And from the cell type deconvolution, we identify that the ApoE44 astrocyte has the most significant enriched matrosome. And those results are predicted same functional module, including chemotaxis, activation, and synthesis of lipids. And is that lipid-related pathway in pure astrocyte microglia pathway enriched in this deconvoluted one? And it was the same case that we do see that lipid metabolic dysregulation along with the matrosome in both microglia and astrocytes. And its predicted function is also the same: increase of synthesis of cholesterol and accumulation of lipids and decrease of efflux. What about the mouse? Because since the field has widely utilized these target replacement mouse of 3.3 and 4.4, when we purify the 3.3 microglia and 4.4, all these genotype primary microglia, and compare ApoE44 versus 3.3, we do see high inflammation interferon signature as well as a matrosome. It's nearly identical both microglia and astrocytes. However, we couldn't find any lipid-related pathway enrichment. However, we do see that lipid accumulation within that matrosome signature. So we further predict looking into all the genes within the matrosome pathway, but we still couldn't identify lipid-related pathway, but chemotaxis or activation, suggesting that these lipid-related dysregulation is specific to human ApoE44 cell. Next, I'm going to briefly validate what we found in this transcriptomic data analysis. So we have selected 2 individuals that share the same common haplotype and convert 4.4 to 3.3 and differentiate to the astrocytes. Before I show the data, I would like to briefly introduce you about what's known in a lipid metabolic trafficking pathway in periphery. So LDL particle, which carries a cholesterol, binds to LDL receptor, internalized through early endosome, sorting endosome, lysosome, and digested lysosome, and communicate with the ER to detect sensing the intercellular level of a cholesterol. When cell has an excessive amount of cholesterol, and free cholesterol will convert to cholesterol ester, and forming a lipid droplet, or it export out to the extracellular space through the ApoE, and ABCA1 is a lipidating machinery. ApoE carries lipids through the ABCA1 and lipid protein is going out. When cells do not have enough cholesterol, free cholesterol is synthesized to the ER through the cholesterol biosynthesis pathway, and also try to uptake from extracellular space and stop the efflux process. What we see in transcriptome is that we have continuously upregulated cholesterol biosynthesis, and it accumulated in a cell, which is rare, and decrease of cholesterol efflux. So we first try to confirm that whether that is the case. So we use a gas chromatography and mass spectrometry and identify that there are 20% increase of total cholesterol, and that pool is derived from free cholesterol. And we further measure the SRBP2 cleave, SRBP2, and HMGCA is increased in ApoE44 cell. These are the rate-limiting enzyme for the cholesterol biosynthesis, and SCAB is a cholesterol sensor. Those are all increased, suggesting that there are de novo cholesterol synthesis is happening. Then it is really good, right? There is still a lot of cholesterol accumulation in a cell. So we chase for where the free cholesterol is localized, and by co-staining with the filatin, which binds upon cholesterol binding, and also the phytidextrin, that's marker for the lysosome, and we see more yellow puncta in ApoE44 cell, suggesting that there are increase of a colocalization in ApoE44 astrocytes. And we further take into the different regions of a brain, Alzheimer's disease human brain, and we found that those phenotypes that colocalizing free cholesterol in astrocyte and lysosome is present in an ApoE44 patient. So what about the lipid carriers? These are one of the cell autonomous effects of ApoE44 that 44 astrocytes, and they see that much less level of ApoE44, both transcriptome as well as the protein level, and intracellular as well as secreted ApoE, and ABCA1 level was significantly decreased, and followed by when we chase for the free cholesterol, it shows a significant decrease in ApoE44 and 2% ATL in the lipid case, as well as methylbeta-cyclodextrin when we try to extract the lipid, and that phenotype is still present. So as a proof of concept, we have treated on LXR-RXR agonist that is increase ApoE and ABCA1 to increase the efflux of cholesterol, and we see that there are increase in ApoE and ABCA1 and ApoE44, and it reverses the efflux phenotype. So the last slide about the matrixome validation. Since we, from the cell type deconvolution, we see the significant matrixome enrichment in ApoE44 cells, so we actually run the Luminex multiplex immunoassay from the all ApoE33 and 44 case, and it was hypercluster showed that genotypes are nicely segregated by these different chemokine, cytokine, and growth factor, and ApoE44 cells show the increase of chemokines, cytokines, as well as growth factor. And interestingly, this signature become augmented and 2.5-fold when astrocyte is interacting with neuron. So it was much more increase in ApoE44 derived from the astrocytes. So last question we asked is what is the cause and consequence relationship with the matrixome and astrocytes? And we treated TNF-alpha interferon gamma, which is widely prevalent in the AD brain. We do see that the lipid synthesis pathways are exacerbated. It was even more increased by the treatment, and decrease is even reduced upon treatment. So we should suggest that there are matrixome literally exacerbate the lipid metabolic dysregulation in ApoE44 case. So in summary, ApoE local haplotype is inherited in an ApoE genotype dependent manner, and this 4 genetic approach of ApoE4 in brain cell type reveal that the cell type specific, human cell type specific dysregulation of cholesterol metabolism in microglia and astrocytes and matrixome in mixed culture as well as AD brain. And in vitro analysis show that elevated de novo cholesterol synthesis is due to lysosomal sequestration in cholesterol, and it is validated in an hippocampal astrocyted AD brain. And in vitro validation also shows that matrixome pathway is associated with the upregulated chemotaxis, glia activation, and lipid synthesis, and that is originated from the astrocyte when it's upon interaction with neuron. So the last part, we are currently working on asking why is this mechanism by which ApoE4 and astrocyte lead to lysosomal sequestration, and understanding by autophagy and defulphagy would run through the multiple different autophagy and defulphagy pathway. And secondly, we have studied at the baseline. Now we are working on towards the how are these different glial cell, ApoE genotype glial cell response to the amyloid challenge, both in vitro and in vivo chimeric model. And lastly, we are working on APOE2 Christchurch and Jacksonville, which is known as a protective variant side of APOE gene. So we hope to identify the interesting pathway related to that. And I would like to thank to my current lab member and as well as a collaborator and my funding source. Thank you. I'll be happy to take your questions.
Thank you. That was an excellent talk. Questions?
Julia, great talk. Your matrosome has a large secretory inflammatory signal. And that's kind of the same thing one sees with the aging, classical aging phenotype too. I was wondering how much of that matrosome is related to like an accelerated aging phenotype and how the aging differs from
I think that's a great question. And I know your group also have been showing the longer aged culture of multiple. So I would say in organoid, if you culture long time, probably matrosome signature probably enriched. And I have heard about that from other colleagues as well. And as you know, that matrosome signature is really huge. And we haven't directly teased apart with the aging versus young versus old, this aging signature. However, when we look at AD, we did APOE33 carrier, which is a negative for APOE4 genotype in AD versus APOE44 case. And we do see much more stronger. So without the APOE4 SNP, it still have a very minor effect by just I would say we didn't compare with young patient. However, within the APOE4 phenotype and APOE3, they have a little bit of matrosome, but upon having APOE4, the signature is much stronger. And we reported actually supplementary that APOE3 patient with just an aged Alzheimer patient also have some matrosome signature range.
Yep. Wait.
So maybe my question is lying because I'm not in your field. So it seems to me the astrocyte, really is critical for the disease pathogenesis. My question is, either you can just get rid of the APOE loss astrocytes or restore APOE4, in the astrocyte. Would that actually change the disease course?
That is a fantastic question, and actually, whole field is trying to figure that out. And we do see APOE4 has gain of toxic function as well as loss of function. And at the baseline, I didn't show it here, but at the baseline, when we remove the APOE4, and it is similar to the APOE4. APOE4 is similar to the APOE knockout, but upon having amyloid, it going to a different route that actually APOE4 and amyloid addition is so toxic that a whole field is that we should maybe reduce it. So actually that's really controversial that in a field that whether, since we are losing the effect in a glial cell, do we have to increase APOE level in a cellular level wise? And in an amyloid disease, the Alzheimer's disease perspective, even Christchurch mutation, which is protective, who carries protective variant, they have a high APOE and high amyloid. So APOE is present near the amyloid, as I showed in the introduction, and that's still present, and might be more related to the tau. So actually, we probably, this is really field in the APOE field that we are actually trying to figure out what's going on in intracellular APOE versus extracellular APOE. So we don't know. We're trying to answer that question. Yes, that's a great question.
Last question, David.
Hi, Julia. Is there any evidence about whether any of the pathways you're studying are strain related? Different strains of the data may activate the pathway, steps, or speaking from a Leon perspective of thinking about strains. There are strains of A-beta.
Yeah, so we have screened all these GWAS. They don't carry APP mutation. So in terms of strain, so maybe I can answer this way. I don't see any difference in strain, but when I look at the postmortem AD brain data, deconvolute all the glial cells looking only neuron, and they have just consistent changes in TCA cycles and mitochondrial defects. But we haven't looking at a single cell level, meaning each individual variability that how these different background would, I would say strain means different individual would affect any downstream. We haven't looked at it that way and looking at each individual case, but
You have to do it like some in vitro experiments.
Right.
So it's
right.
not asking some
Yeah, it's a good question.
Okay, thank you, Julia. And I'll try to keep my closing remarks short because I know it's getting at the end of the day, but I really want to thank everyone that's still here attending this GSI Research Symposium. It's been 2 years of putting up with the pandemic, so I really appreciate all of you coming out. I want to thank our poster presenters that all did an excellent job presenting their posters. The lightning talk speakers today, Anthony, Mandy, Robert, Ahmed, Stephen, our assistant professor faculty, Ziba, Powell, Angie, and Julia here today. Our vendors who supported us in showcasing their work, Checkers Catering for the food, my co-director colleagues, Ala, Grishak, Rachel Flynn, and our admin coordinator, Abby Gatanti over here, who mends our GSI email. If you have any comments, suggestions for how we can improve this symposium, please email us. Also, please reach out to the GSI for the monthly seminars and any other questions you may have about promoting your genetics and genomics research. And thank you again for attending, and hope to see you all next year.